Thank you for that kind introduction. It's good to be back. I think this is my 4th year in a row at Working2Walk. And so I'm going to be talking about two technologies that may be familiar to some of you. Some of you heard me talk before. We'll be talking about ways of controlling scar formation in the injured spinal cord and making the right kinds of astrocytes to promote functional repair, repairing spinal cord. So Wise gave us a very good overview this morning of spinal cord injury. And I'm going to start my talk by just quickly adding to that and talking about some of the pathways in the spinal cord and what is it we're trying to repair and what pathways, what cells might be in pathways we'd like to replace and how we can get nerve fibers to regenerate and form functional connections. There's obviously there's the brain, spinal cord, and there are neurons, as we heard this morning, that sit in the brain, the layer 5 of the cerebral cortex, and they have axons that run all the way down the spinal cord. And as we heard, these are really big cells. And so there are also neurons which sit outside the spinal cord, sensory neurons which send their axons into the spinal cord and relay sensory information up to the brain. So obviously the higher up you have your spinal cord injury, the worse off you are, as well as many of you know, because you now have disrupted the flow of information from the brain down the spinal cord, and the sensory information isn't making it up to the brain. So we look in cross-section, a schematic of the human spinal cord. What we find is that these sensory and motor pathways are segregated. They're found in different parts of the white matter pathways that sit on the outside of the spinal cord. So when someone has a spinal cord injury, a contusion injury, the spinal cord, as you well know, many of you know, bleeds from the inside out. And it bleeds from the inside out because the grey matter contains 5 times the density of blood vessels of white matter. And so there are people walking around who had a spinal cord injury and had a bleed in the center of the spinal cord, and they got away with it because that injury did not impinge out onto the white matter. And it's when you have disruption of those white matter pathways, you have a major loss of function because you've disrupted that flow of information up and down the spinal cord. And then what happens is, as we saw before, the contusion fills with macrophages and fibroblasts, and a scar forms, an inhibitory boundary forms at the edge of the injury site, which stops axon growth. So over the past 100 years, there are 4 main theories that have been put forward to try and explain why it is that the nervous system doesn't regenerate after injury, the spinal cord doesn't regenerate. And the first idea is that adult neurons have lost the ability to regenerate, and there's good evidence to support that. The next is there's a lack of growth factors that as part of normal development, the adult's brain and spinal cord loses growth factors that might help support axon regeneration. Then there's a theory that was proposed about 20 years ago or so that the myelin in the white matter, what makes white matter white, the myelin sheath around the axons actually has molecules on it that are actively inhibitory to axon growth. So now not only have we got a loss of growth factors and maybe changes in neurons, we've also got active inhibition associated with white matter in the pathways. The idea being that these white matter pathways are now becoming a hostile environment to grow axons. And then the last theory, which is one of my favorites, is that scar tissue, which develops directly at the site of injury, presents a combined physical and molecular barrier to axon regeneration. So we've been studying scar tissue in my lab for several years. And this is a study in adult rat, and this is the surface of the spinal cord, and it's a scissor cut to the spinal cord. It makes the worst kind of scar tissue. And we were interested in what cells and molecules were present in the scar that were effectively inhibiting axon growth. And so we have fibroblasts which invade from the outside of the spinal cord and invade the center of the injury site, expressing a molecule called NG2, which some of us think is axon growth inhibitory. There's some debate about it, but I think it's axon growth inhibitory. And there are molecules such as neurocan, it's another chondroitin sulfate protoglycan, CSPG, which is found in the boundaries of the injury site. And then there's other molecules such as tenacin C, and there are subforms of tenacin C which are inhibitory. The take-home message here is that there are multiple axon growth inhibitory molecules that are forming in the scar that axons would then have to negotiate through to be able to regenerate and form new connections. So we went to really high power, as Phil was saying. Scientists like using their microscopes and seeing what's going on in this scar, who's contributing to scar formation. And the idea was that a molecule, neurocan, the idea was in the literature that it's made by astrocytes, star cells, which I'll be talking more about in a minute, the major support cell of the nervous system. And the idea is that they form scar tissue after spinal cord injury. Well, what we were seeing, in fact, was that it wasn't necessarily the astrocytes in red that were giving rise to this inhibitor neurocan in green. we found it was actually glial progenitors, stem cells in the type of stem cells already in the spinal cord that were actually expressing a lot of these inhibitory molecules that were contributing to scar formation. So this gave this insight in 2003, really helped us when we were considering using stem cell technologies for repairing the spinal cord. We had to be very careful putting stem cells, naive stem cells, into spinal cord injuries because there were already stem cells in the spinal cord that were contributing to scar formation. So, the inhibitory environment of the injured spinal cord. This is a spinal cord injury. This is the gray matter, white matter pathways. And if we were able to change that injury site, maybe we were able to suppress scar formation and get axons to grow and make it more permissive. Those axons, these are ascending sensory axons and descending motor control axons. We have a spared axon here that wasn't cut by the injury. These axons here that are trying to regenerate would have to adapt to the injury site, and then be able to adapt to now growing in the environment beyond the injury site. There are lots of different molecules in the injury site that that they have to adapt to be able to grow here, and then they have to adapt to an environment full of myelin. And so it's one thing to get axons to cross an injury site, but those axons won't necessarily now lead to the growing across the injury won't be enough to give you functional recovery. The way axons actually make connections in the spinal cord is that they send out side branches called collaterals that now have to grow from the white matter into the gray matter and form connections with neurons in the gray matter of the spinal cord. And so there's two stages. You have to get the axons to cross the injury and form collaterals. That's how most of the connections are made in the nervous system. And you can have a spared axon here that you would also want to form these new collaterals. In this way, then, a relatively small number of axons can make lots of connections down the spinal cord by sending out these side branches. So, in general, trauma and inflammation after spinal cord injury give rise to scar tissue, and you can see some axons that have grown down a pathway. We have some old experiments where we transplanted adult neurons into adult central nervous system and grew axons down pathways, and they stopped when they entered the glial scar. So trauma and inflammation promotes it's a severing of axons, loss of neurons and glia support cells, formation of axon growth inhibitory scar tissue. And so we're developing two main repair strategies in my lab to promote repair of spinal cord injury. The first is to overcome the scar and the myelin-associated inhibitors. And there'll be a few people surprised to hear me say that. I think, yeah, myelin may actually have axon growth inhibitory molecules in it. And in fact, it does. And the second strategy is to transplant specific types of astrocytes to promote repair of the injured spinal cord. So first I want to talk about astrocytes. Cell-based therapies to repair spinal cord injury. There are a lot of cells to choose from now. And a lot of them are from outside of the central nervous system, CNS, Schwann cells, engineered fibroblasts, macrophages, umbilical cord cells. Now, these cells have varying degrees of effect on the injured spinal cord. And some are having some effects, though I think maybe in some cases we could do better. But what's driving the use of these cells mainly is that they're easy to get hold of. And I would argue that really, in terms of repairing the central nervous system, shouldn't we be thinking about trying to make central nervous system cells? Well, people have thought about this for a while, and so there were groups that were taking immature glial support cells, that would be astrocytes in particular, and transplanted them into spinal cord injuries, a little bit of axon sprouting, no functional recovery, rather disappointing. Then there was groups that took neural stem cells. Once again, saw some benefits, but we think we could do a lot better. And glial progenitors, as I say, a subtype of stem cell that people have tried, and there's varying degrees of effect, and most labs aren't seeing robust recovery. So, which is the right cell type to try and repair the injured nervous system? Which is the right cell type to repair the injured spinal cord? Well, we had seen in our studies that there were astrocytes that were forming scar tissue, stopping the axons in green from growing. And yet there were also astrocytes in red that were supporting the growth of axons away from spinal cord injuries. So even in the adult injured spinal cord, there seemed to be two different types of astrocyte at least. Some were supportive of growth, and some are inhibitory. So the majority of the human central nervous system is made up of astrocytes. The idea is that 70% of all cells in the human brain and spinal cord can be classified as some type of astrocyte. And so in thinking about using astrocytes for repair, in particular, can we make astrocytes that are axon growth supportive? Are astrocytes actually good candidate cells for repairing the injured spinal cord? What do we know about astrocytes that makes them a good cell to think of using for repair? Potential benefits of transplanting the right astrocytes. It's long been known that astrocytes provide metabolic support for neurons. One idea is that they sit between neurons and blood vessels and take metabolites from the blood vessels and basically provide, say, support for neurons. The idea is also they maintain the blood-spinal cord barrier, which is broken down after spinal cord injury. As I'll show you in a minute, if you get the right kind of astrocyte, they're incredibly good at bridging spinal cord injuries and promoting axon growth. They're also known to regulate remyelination, that forming those new sheaths. We heard that Geron's very interested in doing this with their glial progenitors. Well, it's now known that also that astrocytes control how progenitors in the normal spinal cord, they control the differentiation of those progenitors into myelinating cells, oligodendrocytes. They're also been shown recently to release factors such as thrombospondins that promote synapse formation, the connections between neurons. They also have their own networks. We're now realizing that we've been too neuron-centric. We discovered neurons and electricity about the same time, about 100 years ago, and so we thought that the nervous system was wired just like our house. And to a certain extent, yes, axons are very important, but we were ignoring what the astrocytes were up to. It's now realized that they have their own signaling networks and are signaling amongst themselves with calcium waves and other signaling mechanisms. What's really exciting is that we're now realizing that astrocytes control the activity of synapses. So I've just told you they promote the formation of connections, synapses between neurons, and now it's realized that they also regulate the activity of those synapses by releasing their own gliotransmitters. You've heard of neurotransmitters. We're now realizing that astrocytes can release their own factors, transmitters, into the gap between the two neurons and regulate the activity by either suppressing or promoting activity of synapses. So I would argue, and a lot of us in neurobiology are now realizing this, that astrocytes are essential components of brain and spinal cord neural networks. So if we're serious about repairing the injured spinal cord, we really need to be thinking about astrocytes because there are a lot of things that they can do. And we want to put in cells that not only release growth factors or maybe make a bridge, but cells that can actively participate in the function of neural networks if they remain in the spinal cord, if they survive after transplantation. Can we make the right kind of astrocyte for spinal cord repair? So, embryonic stem cells in themselves, we don't think are that good necessarily for repairing tissue. And they don't build tissue straight off. They have to give rise, a pluripotent cell would have to give rise to more restricted types of stem cells that just make nervous system, or stem cells that just make liver. Because we're interested in making nervous systems, so we're interested in neuroepithelial stem cells. And we know that these cells can give rise to precursors that just make neurons, and also give rise to precursors that just make glial-restricted precursors. Now, we're interested in making astrocytes, so obviously we're interested in glial-restricted precursors. And these cells, we can extract them from fetal tissue. We've actually been able to do this not only for rat, but also from human now. That work is ongoing, can't talk about it today. But what we do know is that if you then, you can treat these glial precursors and turn them into oligodendrocytes. And if you give them two classic signaling pathway molecules, astrogenesis pathway, signaling molecules, bone morphogenetic 4 or ciliary neurotrophic factor, you can drive these glial precursors to making two distinct subtypes of astrocytes. So I think I'm right in saying that we're one of the, probably maybe the first group working with the University of Rochester, New York, to be able to make defined different populations, different types of astrocyte. They look very different, and they have very different characteristics, as you'll see in a minute when we transplant them into the injured nervous system. So we can make pure populations in just 7 days, two different types of astrocyte. What's interesting about the GDA's BMP, that's glial-restricted precursor-derived astrocyte by the BMP pathway. It's quite a mouthful, but it really describes which specific astrocyte we've made. These cells have low levels of the molecules we find in scar, the CSPGs, and are highly supportive of axon growth, whereas the GDA CNTF have high levels of CSPG and are somewhat like the astrocytes we see in scar tissue. So obviously we're interested in making these cells. Now the big question is, can we make the beneficial GDA's BMP, these astrocytes here, from other stem cell sources? And we're excited to hear about Phil's work, and it might be fun to collaborate in the future, and, because we think the GDA's BMP is a very exciting cell. So take the GDA's BMP, and actually I will be showing work with some of the other astrocytes too, transplant them into spinal cord injury in rats. Do they promote axon regeneration? Do they promote functional recovery? experimental spinal cord injury model. It's adult rats we're working with, 3-month-old, C1, C2, in this case, transection injury. It's a nice injury for testing. Are these cells good at making bridges? How well do axons cross the injury site? We put the cells in, and then we can add a tracing molecule called BDA, and then look to see whether these axons can regenerate across an injury bridge with these different cells. And what we're finding is the GDA's BMP are remarkably good at bridging spinal cord injuries. Here's the injury site outlined here with the dotted lines. The red cells, the transplanted GDA's BMP, axons in green. And if we look at the efficiency of regeneration across the injury, if we have 100% of the axons before the injury, we found 60% of the axons made to the center of the injury, and on average 39% beyond the injury. The previous record was with marastromal cells from the Tuzinski group. They got 5% axon growth across the spinal cord injury in just 1 to 3 months. We saw 39% in 8 days, just with the right kind of astrocyte. If we took the naive stem cells, the GRP stem cells we used to make the astrocytes, put them in a spinal cord injury, they didn't make a bridge. I hope you can see the axons aren't crossing the injury site here. In fact, what we found was that the transplanted naive stem cells were turning into scar-like astrocytes at the injury site, and nerve fibers weren't able to cross the injury. So it wasn't enough to put in the glial precursors, that the embryonic type of stem cell that we used to make the astrocytes. They didn't work. If we made the other type of astrocyte, the GDA-CNTF, now you would think that any kind of astrocyte you can make from an embryonic stem cell would be really good. But no, what we're finding here is that the cells fill the injury site, the axons won't grow across, and here's another animal that's got GDA's BMP with great axon growth. And if we compare the efficiency of growth across the injury site with no treatment at all, we saw 4% axon growth. If a lab tells you they're saying 0 growth across an injury, they're not doing careful analysis. You always see some axons cross an injury. What we saw then, where we put the precursors in, next to no difference, the stem cells. The GDA-CNTF, once again, very little difference, 6% across the injury. In our hands, not statistically significant. You get the right kind of astrocyte, a major effect on axon growth across the injury. What about functional recovery? We've been making injuries of the cerebrospinal tract, which is a pathway that controls targeted movement of pores. We're interested in looking at brain control of spinal cord movement, and because that's really important for humans in terms of being able to control your limbs and be able to walk. So we cut this particular pathway in rats, and we could then count how many mistakes these rats make crossing a ladder. It's a very stringent test of brain control of movement in rats, the grid walk analysis or horizontal ladder. And what we found was that before injury, these rats are making 1 to 2 mistakes per run. We give them an injury, transplant either the stem cells in or the GDA's BMP, and down is good, up is bad in terms of how many mistakes are being made. So by 3 days, the animals that receive the right type of astrocyte are making half the number of mistakes in just 3 days after receiving these cells. And 8 to 9 animals per group, and by 14 days, they're back to normal scores, and no recovery with those animals that receive the glial precursors, the stem cells. In fact, you can take these animals out many months and they don't recover. If you make the the other type of astrocyte, the GDA-CNTF, remember, up is bad, down is good. Once again, no difference from control like the stem cells, and yet here's another set of animals with robust recovery with the right kind of astrocyte. So I want to quickly talk about pain as a nasty potential side effect of cell-based therapies for repairing spinal cord injury. This was a great paper from Lars Olsen's group, which showed that allodynia, pain syndrome, was an unfortunate side effect of using human neural stem cells in adult rat spinal cord injury. And then saw that the neural stem cells turned into astrocytes, and this promoted pain. This was a worry for those of us trying to use astrocytes to repair the spinal cord. It's all very well getting some functional recovery, but if you have a nasty side effect, such as severe pain, then that could be debilitating in itself. So we knew that the neural stem cells had to turn into GRP stem cells before they could make an astrocyte. And so what we did was basically, there's a lot of data here, but we're looking at induction of pain weeks after injury in animals that received either the right type of astrocyte, the second type of astrocyte that can give us any functional recovery, or the stem cells. And what we're basically seeing is that the second type of astrocyte and the glial precursors are both promoting pain here, whereas, lucky for us, the GDA's BMP are giving all that functional recovery and not inducing pain. So you can get functional recovery, good regeneration, without having induction of sprouting of pain circuits and pain problems. So quickly, in the last minute that I've got, I'm going to quickly talk about Decarin. So we've heard that there's a problem with scar tissue. How can we regulate scar in the central nervous system? How can we prevent it from forming, or how can we break it down? And this was an experiment where we had been infusing a molecule called Decarin. It's a naturally occurring molecule that's a potent suppressor of scar formation, as we have found, and others have seen too. And it's found throughout the body, and it's made in the spinal cord by astrocytes, and it's also made by motor neurons, make a lot of Decarin. So we could infuse Decarin into a spinal cord injury. It's a lesion center, injury center. Look at the ability of these neurons to cross the injury site, and now instead of stopping at the scar's been suppressed with Decarin infusion. The astrocytes in red are not walling off the injury site, and axons are crossing the injury site in just 4 days. And so we actually quantified how much we had suppressed the scar, looked at different inhibitors, and found some of them down by almost 90%. So it had a major ability to suppress scar formation. And I'm pleased to say, rather, we won the Asia Award 2006 for breakthrough in research for our work with Decarin. Can Decarin break down scar tissue? Well, we know that there's an enzyme called plasmin, which has the ability to break down scar, and it's found throughout the body, and usually used by the body to modify scar tissue. And we saw a 17-fold increase in this enzyme in Decarin-treated spinal cord injuries. What about chronic spinal cord injury? Well, we're one of the few labs that's also studying chronic injuries in rats. And 9 months after injury, unfortunately, the scar tissue is now spreading up the tract. There's more of a challenge, as we all know, in regenerating the chronically injured spinal cord. So how can Decarin help? One means might be to break down scar tissue. And we've got promising new data to suggest that the enzymatic approach that Decarin may be doing just that. But the other mechanism would be to desensitize the neurons to the inhibitors found in the scar, to myelin and the CSPGs. And so we conducted an experiment where we took adult sensory neurons, put them onto myelin that we find in the white matter pathways, and indeed there was very poor outgrowth, as these neurons are sat here, they're not growing axons. If you had Decarin, you got an almost 5-fold increase in axon growth in 16 hours. Labs are happy with 50% increase. We saw almost 5 times the length of growth. If we took the neurons and we put them onto high levels of the CSPGs we could find in central nervous system scar tissue, they don't want to grow. You add Decarin, you got an almost 15-fold increase in axon growth in just a matter of hours. So in addition to being able to suppress the scar, to lower the hurdle in front of axons when they're trying to cross an injury site, Decarin also has the remarkable ability to desensitize the neurons to the inhibitors and boost their ability to grow in environments such as the chronically injured spinal cord, where we know there's high levels of CSPG and still myelin debris hanging around. So in summary, I hope I've convinced you that GDA's BMP and Decarin, I think, are really highly promising technologies for acute and chronic spinal cord injury. Controlling the fate of stem cells is required to optimize repair of spinal cord injury. Putting stem cells into injured tissue and hoping that somehow the stem cells know what to do and the injured spinal cord knows how to use them, I think is hoping for a little too much. And that what we're seeing here, of course, is that if you don't control what the stem cells do, in fact, what they can do is turn into scar and actually also promote pain syndromes. And making the right kinds of astrocytes, I think, should be a major focus of stem cell technologies in the future. It's really embarrassing as a neuroscientist to stand here in front of you and tell you how little we know about astrocytes, different types of them, yet they make up 70% of all cells in the central nervous system. Ongoing studies. Testing, we're currently testing human Decarin and human GDA's BMP in chronic contusion SCI models. We're looking at combinations of the GDAs and Decarin and how you combine them with rehab in the most effective way. These 2 lines of research were always designed to come together, and we're now doing the combination experiments. And we are in the process of developing GMP-grade human GDAs BMP for future clinical trials. Thank you very much.
Visit u2fp.org for more coverage from Working to Walk and the latest in spinal cord injury news.